Hi everyone, welcome back to Pill Talk. Today we will be discussing into an incredible topic that is muscle physiology. So muscles are fascinating structures that power every move we make. So by the end of this session, you will understand how they work, how they are classified, and the secrets of their molecular mechanism. So without any delay, let's begin. So the muscles are the engine of our body. You know that muscle cells have the unique ability to convert chemical energy into the motion. So this process dominates muscle cells. It makes them essential for our survival. Simply, if we're going to define the muscles, then muscles are the long cells that contract when stimulated. It generates the force and movement needed for internal regulation and external activities. Simply, we can say a body tissue consisting of long cells that contract when stimulated and it produces motion. If we classify the muscles, then muscles are classified into various types on the basis of three factors. Number one is presence or absence of striations. Number two is control. Number three is location. So the presence or absence of striations. Think of striated muscles like a finely striped shirt, while non-striated muscles are smooth and plain. And if we talk about control, voluntary muscles move when we want them to move, while involuntary muscles work on their own. If we talk about location, or we can say situation, skeletal muscles support bones, cardiac muscles power the heart, and smooth muscles are found in internal organs. So together they ensure our body functions seamlessly. You can see through this table, it definitely perfectly classified the muscles based on striation, based on control, and based on situation. So you can take a screenshot or screen snip of that table so that you can read out later. Next is the structure of skeletal muscle. So let's explore the anatomy of skeletal muscle. Each muscle is made up of bundle of muscle fiber. So these bundles are wrapped in connective tissue for strength and flexibility. Epimysium covers the entire muscle. Perimysium wraps each bundle or fasciculus. Endomysium protects individual fibers. Think of it as a well-organized team with each member contributing to the bigger picture. Muscle mass or muscle tissue is made up of large number of individual muscle cells called myocytes, or we can also say them as muscle fiber. And the muscle mass is separated from the neighboring tissue by a thick fibrous tissue layer known as fascia. And all the coverings I have already explained. So let's take a closer look at the structure of a skeletal muscle, which is beautifully illustrated in this diagram. Starting from the connection to the bone, muscles are anchored by tendons. Tendons are the tough bands of connective tissue that transmit the force generated by muscle contraction to the bones, enabling them to move. So now let's explore the layers of the muscle. So epimysium, this is the outermost layer, a connective tissue sheath that wraps around the entire muscle, providing protection and structural integrity. Inside the muscle, you will find bundles of various groups like fascicles, which are surrounded by another layer of connective tissue called perimysium. And each fascicle contains individual muscle fiber, which are also called as muscle cells, which are encased in a delicate connective tissue called endomysium. You will also notice the presence of blood vessels, which play a critical role in supplying oxygen and nutrients to the muscle fiber and removing waste products. So this intricate organization ensures muscles can perform efficiently, whether lifting a weight or just holding a posture. So the layers epimysium, perimysium, and the endomysium work together to provide strength, flexibility, and protection while allowing the muscle to function effectively. It's a remarkable example of how structure meets function in the human body. Let's discuss muscle fiber under the heading of structure of skeletal muscle. So if we discuss about its shape and size, muscle fibers are cylindrical in shape with an average length of 3 cm. So this can vary between 1 cm and 4 cm depending on the specific muscle. And the diameter ranges from 10 micrometer to 100 micrometer, a size that contributes to the strength and flexibility. If we talk about the connection to tendons, so these fibers are attached to tendons, which are tough cords of connective tissue, as I already discussed. In some cases, the tendons are thin, flat, and stretch, forming a structure called aponeurosis, which is just as strong and functional. If we talk about protective layers, each muscle fiber is encased by a plasma membrane referred to as sarcolemma, which lies beneath the endomysium, which is a connective tissue layer. If we talk about its internal environment, inside the sarcolemma is a sarcoplasm, the cytoplasm of the muscle fiber. So this is where vital cellular activities occur to enable muscle contraction and energy production. We are going to discuss about some structures embedded within the sarcoplasm are number 1 nuclei, number 2 myofibrils. We are going to discuss them later. Number 3 Golgi apparatus, number 4 mitochondria, number 5 sarcoplasmic reticulum, which is a specialized endoplasmic reticulum that stores and releases calcium ions crucial for muscle contractions. Number 6 ribosomes, number 7 glycogen droplets. They serve as a reservoir of energy by storing glucose, which is actually critical during muscle activity. Number 8 lipid droplets. They are occasionally found. These provide an additional energy source during prolonged muscle activity. Now let's explore the myofibrils, which are the functional units of muscle fiber and they are vital for muscle contraction. So the question arises, what are myofibrils? These are fine parallel filaments located within the sarcoplasm of the muscle cells. So myofibrils are responsible for the muscle ability to contract and generate force. If we talk about its structure and length, myofibril extend throughout the entire length of the muscle fiber. It ensures coordinated contractions. If we talk about how they look under the microscopes, so when we take a cross section of a muscle fiber, myofibrils appear as a small dots scattered within the sarcoplasm. In some cases, these myofibrils group together into patterns known as Conham areas, or we can also say that Conham fields. Let's discuss about the microscopic structure of a myofibril. So under a light microscope, each myofibril shows a pattern of alternating band. These are also known as sections, segments, or discs, and are formed by muscle proteins. So there are two types of bands, light band or I band. Number 2 is dark band or A band. Now let's break down the light and dark bands in skeletal muscles, which gives them striated appearance. Number 1 is light band or I band. So the I band is called isotropic because it is isotropic to polarized light. This means when polarized light passes through this area, it refracts at the same angle, resulting in uniform light appearance. So it contains thin actin filaments only, making it less dense and lighter under a microscope. Number 2 is dark band or A band. The A band is also called as anisotropic or anisotropic because it is anisotropic to polarized light. Here, light rays are refracted at different angles, giving it a darker appearance. So this band contains thick myosin filaments, often overlapping with thin actin filaments, which makes it denser. Interestingly, the A band is also referred to as the Q disc, derived from the German word Querschiebe, meaning the cross disc. And the diagram illustrates how these bands are arranged within a muscle fiber. So the sarcolemma encases the muscle fiber, which contains myofibril displaying alternating A and I bands. The bands contribute to the organized striped appearance critical for muscle contraction. So these bands are fundamental to how muscle contract, and we will discuss further in the next slides. Now let's discuss the Z-line and sarcomere, which are integral components of skeletal muscle structure. So the I band previously discussed is divided into two portions by a narrow and dark line called the Z-line or Z-disc. So the term Zwischenschiebe in German translate to between the disc, emphasizing its position. So the Z-line is formed by a protein disc that doesn't allow light to pass through it, so it gives it a distinct appearance. And next is the sarcomere. The sarcomere is the portion of a myofibril located between two Z-lines. It is considered the basic structural and functional unit of muscle contraction. So when muscles contract, the sarcomere shorten. It brings the Z-lines closer together while maintaining the alignment of the bands. As you can see in the diagram, the Z-line is clearly visible as dividing line within the I band. So the sarcomere spans from one Z-line to the next. It encompasses the alternating bands. Let's take a closer look at the sarcomere, which is the fundamental contractile unit of a skeletal muscle. A sarcomere is defined as the structural and functional unit of skeletal muscle. It is also known as the basic contractile unit, where the actual process of muscle contraction takes place. So the key components are here. Each myofibril is made up of alternating dark A band, anisotropic, and light I bands, isotropic. So within the A band, there is a lighter region called the H zone, which is visible when the muscle is relaxed. In the middle of the H zone, you will find the M line formed by myosin binding proteins. So the M line helps stabilize the arrangement of myosin filaments. In the diagram explanation, it shows the structure of a sarcomere, which spans from Z line to the next. You can see how the A band and I band, H zone and M line are arranged to ensure the proper alignment of filaments for contraction. Let's discuss into the electron microscopic structure of the sarcomere, which provides a detailed view of its components. So the sarcomere is composed of many thread-like structures called myofilaments, which are essential for muscle contraction. These myofilaments are classified into two types. Number one is actin filament and number two is myosin filament. As you can see through the diagram how these filaments are organized within a sarcomere, the actin filaments extend from the Z line towards the center. The myosin filaments are located in the middle and overlap within the actin filaments in the A band region. The H zone seen in the middle contains only myosin filaments when the muscle is relaxed. Actin filaments are also called as thin filaments and they are anchored to the Z line. And myosin filaments are also called as thick filament and they are positioned centrally in the sarcomere. Now let's discuss the role of cross bridges in muscle contraction, which are vital components of the sliding filament mechanisms. So the question arises, what are cross bridges? Cross bridges, also called as myosin heads, are lateral projections that arise from each myosin filament. At the tip of these projections are myosin heads, which are in large structures specifically designed to interact with actin filaments. So how do cross bridges work? These myosin heads attach to binding sites on the actin filaments. So during contraction, the myosin heads pull the actin filaments inward, which shortens the sarcomere and it brings the Z line closer together. So this process of pulling actin filament is termed as the sliding mechanism, or we can say ratchet mechanism. It is powered by the hydrolysis of ATP, which provides the energy for myosin heads to attach, pivot and detach rapidly. So the diagram highlights A, the sarcomere in a relaxed state with visible H zone and I band, and the B represents the sarcomere during contraction where the H zone and I band reduce as the actin filaments slide inwards while the A band remains unchanged. And this slide provides a detailed visual representation of the structure of skeletal muscles, from the larger muscle fiber to the microscopic components responsible for contraction. And as you can see, the muscle fiber is composed of numerous cylindrical structure called myofibrils. So these myofibrils run the entire length of the muscle fiber and are responsible for its striated appearance and contractile functions. And then you can see bands and sarcomeres. So within the myofibrils, you can see the I band, A band, and the Z lines, which defines the boundaries of each sarcomere. And the sarcomere is the smallest functional unit of the muscle. It is the location where the process of contraction occurs. And as you can see, filaments within the sarcomere. Thick filaments made of myosin located in the center of the sarcomere, and the thin filaments composed of actin anchored at the Z line extended into the A band and overlapping with myosin filaments. And then you can see cross bridges. The zoom in section at the bottom shows the interaction between the thick and thin filaments. And cross bridges formed by myosin heads interact with actin filaments to generate force during contraction. So this hierarchical structure from muscle fibers down to the contraction of actin and myosin filaments, it showcases how skeletal muscles achieve precise and powerful contractions. Now let's talk about the contractile elements of skeletal muscles, which are the proteins responsible for muscle contraction. Number one, myosin filaments. Myosin filaments are formed by myosin molecules. These thick filaments are equipped with myosin heads, which form cross bridges by attaching to actin filaments during contraction. Number two is actin filaments. So actin filaments are thin filaments composed of three types of protein. Number one is actin, which is the main structural component. Number two, tropomyosin, which is the regulatory protein that covers the binding sites on actin in a relaxed state. Number three, troponin, another regulatory protein that binds calcium ions and controls the movement of tropomyosin. And the interaction of these four proteins work together to form the contractile elements of the muscle. So this intricate interplay is essential for the sliding filament mechanism, which underpins muscle contraction. Now let's focus on the myosin molecule. Each myosin filament consists of approximately 200 myosin molecules. So these molecules are aligned in such a way that the myosin heads protrude outward, ready to interact with the actin filaments. While there are about 18 classes of myosin identified in the human body, only myosin 2 is present in the sarcomere. It plays a critical role in skeletal muscle contraction. So the diagram highlights the myosin head, which has an ATP binding site and an actin binding site. So the ATP binding sites, this site provides the energy required for the myosin head to perform its power stroke, and another site which is also called as actin binding site. So this allows the myosin head to attach to actin filaments and generate force through the sliding filament mechanism. Coming towards the actin molecule, each actin molecule is called F-actin, a filamentous structure formed by the polymerization of smaller units known as G-actin. So this arrangement provides the thin filament its structural and functional properties. Each actin filament is composed of approximately 300 to 400 actin molecules, which aligns in a helical structure. So in each molecule, there is an active site to which the myosin head attaches during muscle contraction. Actin filaments are associated with two important regulatory proteins. Number one is tropomyosin and number two is troponin. So the first diagram illustrates the structure of a myosin molecule, highlighting the heavy and light chain, and the second diagram shows the arrangement of actin filaments along with tropomyosin and troponin. It emphasizes the role in regulating contraction. Let's discuss the role of tropomyosin and troponin. These are the two key regulatory proteins associated with actin filaments. Tropomyosin. So in the relaxed state of muscle, tropomyosin molecules cover all the active sites of actin molecules. So this prevents the binding of myosin heads to actin. It keeps the muscle in a state of rest. Another molecule is troponin. Troponin is a complex protein made up of three subunits, each with a specific function. Number one is troponin I. It is attached to F-actin and it inhibits myosin binding. Number two is troponin T. This subunit binds to tropomyosin. It secures the troponin complex in place. And number three is troponin C. It binds calcium ions. It initiates a cascade that leads to muscle contraction. Let's discuss the molecular mechanism of muscle contraction, which is centered around the interaction of thick and thin filaments within the sarcomere. So the term contraction refers to the activation of the force-generating sites in muscle fiber known as cross bridges. So these cross bridges form when myosin heads attach to actin filaments. Sliding filament mechanism. So this is the primary mechanism responsible for muscle contraction. The thick filaments, which are myosin, and thin filaments, actin, slide past each other within each sarcomere. This movement is propelled by the repetitive binding and pulling action of the cross bridges. So importantly, the lengths of the filaments themselves do not change. Instead, the sarcomere shortens. It brings the Z line closer together. Let's continue with the sliding filament mechanism, which is a fascinating process that explains how muscle generate force and movement. So the ability of muscle fiber to generate movement relies on the interaction of two contractile proteins, myosin in the thick filament and actin in the thin filaments. So myosin composed of two globular heads, one binds to ATP, the other binds to actin, and in the actin it has some specific binding sites for myosin heads, and the energy provided for this process is by the hydrolysis of ATP. And if we talk about mechanism, so the diagram shows the stages of the sliding filament mechanism. So in the relaxed state, each zone is visible and the Z lines are further apart. Myosin heads are not bound to actin. In the contracted state, as you can see, the Z lines move closer as the myosin heads pulls the actin filaments inwards, and the sarcomere shortens, but the lengths of the filaments themselves remain unchanged. And the energy-driven movement is provided by the ATP. Here's a detailed look at the cross-bridge mechanism. Cross bridges are projections from the thick filaments that attach to the thin filament. So this attachment occurs at specific binding sites on actin. It initiates the contraction process. So once bound, the cross bridges undergo a conformational change. It pulls the thin filaments towards the center of the sarcomere. So this action shortens the sarcomere. It reduces the distance between the Z line. So this diagram exactly illustrates that. So the cross bridge movement as it pulls the actin filaments inward, and the continuous cycle of attaching, pulling, and detachment that enables contractions. You should note that while the sarcomere shortens, the filaments themselves retain their lengths. So each thick filament has approximately 200 cross bridges, and only 2 are shown here. So the synchronized action of these cross bridges generates the force required for muscle contraction. And this slide provides a closer look at the structure and function of the thick filament and its cross bridges. So the panel A shows the thick filament composed of myosin molecules. So the myosin molecules are oriented in opposite directions on either side of the filament, with their heads protruding outward to form the cross bridges. And if we talk about the B part, it breaks down into the components of our myosin molecules. They have heavy chains, they have globular heads. One contains an ATP binding site, the other contains actin binding site, and it has some light chains. It support the globular heads and contribute to their function. And the cross bridge formation these cross bridges are formed when the myosin heads bind to actin during muscle contraction, as we already discussed. So this binding is powered by the energy released from ATP hydrolysis at the ATP binding site. Coming towards cross-bridge cycle. So the cross-bridge cycle refers to the series of steps that occurs between the binding of a myosin head to an actin filament and its detachment, and it's being ready to repeat the process. So this cycle ensures the continuous sliding of filament of our muscle contraction. So the diagram illustrates the 4 key steps. Number 1 is high energy attached stage. The myosin head in a high energy attached stage binds to site on the actin filament. Number 2 is power stroke. So as the myosin head pivots, it pulls the actin filaments towards the center of the sarcomere. It shortens the muscle. This is known as the power stroke, and it releases energy stored in the myosin head. Number 3 phase is detached stage. After the power stroke, a new ATP molecules bind to myosin head, causing it to detach from the actin filament. And number 4 stage is reactivation. So the ATP is hydrolyzed into ADP, and inorganic phosphate. It re-energizes the myosin head and prepares it to bind to a new actin site. It will restart the cycle. Now the cross-bridge cycle consists of 4 steps, as we already discussed: attachment, movement, detachment, and energizing. So this diagram provides a detailed representation of the cross-bridge cycle. It highlights the chemical and mechanical changes that derive muscle contraction. Let's discuss in a more simpler form. This slide presents the chemical equations for the cross-bridge cycle. It breaks it into 3 key steps. Step number 1 is actin binding. So the interaction begins with myosin M in its energized state bound to ADP and inorganic phosphate ion. So the equation is A plus M star dot ADP dot inorganic phosphate ion. Here A represents actin and the star asterisk on myosin indicates its high energy state. This represents the binding of myosin to the actin filament, forming a cross bridge. Step number 2 is cross bridge movement, which is also known as power stroke. As the power stroke occurs, the myosin heads pivots, pulling actin towards the M line. So the equation changes to dot M plus ADP plus inorganic phosphate ion. During this process, ADP and inorganic phosphate ion are released, and the energy stored in the myosin head is used to generate force. Step number 3 is detachment. A new ATP molecule binds to myosin, causing it to detach from actin and reset. So the step breaks the cross bridge, allowing the cycle to restart with another actin site. This step ensures the cyclic interaction between actin and myosin powered by ATP hydrolysis. Let's discuss an interesting phenomenon known as rigor mortis, which occurs after death. So what is rigor mortis? The term rigor mortis is derived from Latin. Rigor means stiffness and mortis means of death. It refers to the stiffening of the body that begins a few hours after death, typically becoming noticeable around 2 to 6 hours post-mortem. So the mechanism of rigor mortis is after death, the concentration of ATP declines in muscle cells. So ATP is crucial for the detachment of myosin heads from actin filaments. Without the ATP, non-energized cross bridges remain attached to actin, but movement and detachment cannot occur. It leads to sustained contraction and stiffness. So this results in the stiffened state of the muscles observed in rigor mortis. So the stiffness of rigor mortis typically lasts for about 48 to 60 hours after death. It disappears as the tissue begins to degrade due to enzymatic and microbial process. So the rigor mortis is a post-mortem change that highlights the dependency of muscle relaxation on ATP. And with this, thank you so much for watching Pill Talk. Stay healthy and stay informed. Don't forget to like, share and subscribe.